Good morning to everyone, or good evening to everyone. So, very welcome to geneXplain web seminar today. And it is my very pleasure to introduce our invited speaker for today, Dr. Cecilia Marcondes, who is presently working as Associate Professor at San Diego Biomedical Research Institute. And present research task of Cecilia and her group is to study disorders of the central nervous system with special focus on HIV infection. And while analyzing expression data, Cecilia and her group apply several different bioinformatics tools to analyze their data, different software and databases, and TRANSFAC database is one of such tools for data analysis. We at geneXplain are very thankful to Cecilia for her kind agreement to give a presentation here, present some results of their research here in the frame of geneXplain web seminar. Cecilia, you are very welcome to start your presentation now. Please take over. I will stop and you can share your screen.
Thank you. Good morning, everybody. So I really appreciate the invitation. This is a wonderful opportunity for us to tell everybody about the work that we are performing at the San Diego Biomedical Research Institute here in San Diego, California. This work is supported by the National Institute of Drug Abuse. And this particular part of the work, we are interested in examining human brain specimens, which are a very, very precious resource. Post-mortem specimens are very rare. And we are thankful to the donors that we can look at those brains and learn what's going on. So now, because I know that this audience is quite diverse, I will give a little bit of a background, starting a little bit from the basics, and let's see if I move on. So the subject of our interest is neuro-HIV. So neuro-HIV is a number of symptoms, neurological consequences that happen as a result of HIV, human immunodeficiency virus infection. And they happen because the virus enters the central nervous system really soon after infection, within a couple of weeks. And the way the virus enters in the brain is through infected cells that go in and out across the endothelial barrier that separates the blood from the central nervous system. And these cells go in and out in a surveillance mode, or just as a result of encountering some insult or little things that they have to clean up. And but if they are infected, particularly the monocytes that become macrophages in the tissue, the virus finds the opportunity to jump into cells that are living in the brain. So in this particular case, we have microglia cells. CD40 cells also can do that to a lower extent, but they can. And there are reports that other cells in the central nervous system can get infected as well, although not productively. And in addition, peptides, proteins from the virus that are released in the environment, for instance, the transactivation of transcription TAT protein can be secreted in the environment and have effects in all the different cell types in the brain microenvironment, producing dramatic changes in the molecular landscape and moving towards phenotypes that favor inflammation. So what is important is that this generates cognitive issues, several different levels of neurological consequences, and they persist in spite of antiretroviral therapies. This is something that happens in a very significant proportion of individuals that are infected with HIV. And this is because antiretrovirals have either they have a very poor penetration into the central nervous system, or if they do, they themselves have neurotoxic effects. So another issue is that in the central nervous system, when the cell infects, when the cells that are infected have low replication rate or low activation state, the virus can integrate into the genome, and this makes the central nervous system a safe haven, and the virus uses the cells as reservoirs. So now there are disorders. And what these two conditions, age and substance use disorders, have in the context of HIV infection is an ability to modify the disease. This is largely because of changes in neurotransmitters inside of the central nervous system. So age is likely to decrease the amount of neurotransmitters in the central nervous system, and substance use disorders, on the other hand, will enhance the level of neurotransmitters in the central nervous system. And dopamine is one of the neurotransmitters that is mostly involved in this imbalance of neurotransmitters. And what is most important for us is that the target cells of HIV, the cells that become infected with HIV, immune cells, innate immune cells, macrophages, microglia, and also T cells, they express the receptors for neurotransmitters. So before, we used to have this thinking that cytokines are for immune cells and neurotransmitters are for neurons. And nowadays we know that this is a cross communication. Neurotransmitters can act on immune cells, and cytokines can also act on neurons. So you have a cross communication in the environment of the central nervous system, with dopamine as a central component in this relationship. Now, because this is something that people don't know very much about, is kind of a new idea, we need to take advantage of several different models to understand that better. And I'm going to try to show you that our lab uses several different models to get to understand what we need to understand in this context. So these are a few of the papers of our lab. All of them have something to say about the overlap between substance use disorders and HIV neurological disorders. And though you can see here on the side that we look at all the different models. Each model can answer a different specific question, so all the models have some value. And what I highlighted here are the publications that use TRANSFAC, the GeneXplain platform, to perform part of the analysis. And the yellow one is the one that we are going to talk the most about today. So why do we focus on substance use? Because substance use and HIV have a high overlap in terms of population. And this is because stimulant drugs and also injectable drugs pretty much increase risk behaviors that are associated with exposure to HIV. So not only the stimulant drugs modify behaviors, but also they modify molecular mechanisms that make the cells more susceptible to HIV infection. So for instance, here from the monkey model, we can see that animals that are treated with one of the stimulant drugs that we focus on, methamphetamine, which is highly prevalent in HIV populations, increases the expression of one molecule that is very important for HIV to enter into the cell, which is CCR5. So what we see here is that methamphetamine alone increases CCR5 expression on target cells. The infection, which in the monkey is caused by the simian immunodeficiency virus, a cousin of HIV, also increases CCR5. And when you have the combination of infection and drug use, you have with viral peptides, you have also an increase. And what is interesting for us is that dopamine, which is the neurotransmitter that is released in the brain in the context of stimulant drugs such as methamphetamine, also increases the expression of CCR5. So now this is just for you to have an idea how it looks like in terms of pathology. This is in the monkey frontal cortex, brain frontal cortex. So you have the brown spots are CCR5 positive cells. This is in a situation of infection. You can see an increase, a drastic increase. And here in this, we have two examples of combined infection and drug treatment, where you have an even higher number of cells. Here we have multinucleated giant cells, which are likely associated to the presence of the virus. And also in correlation with that, you have increased viral load in the brain of animals that are infected and also treated with the drug. And also in human cases, this is to show you a human case of HIV-infected drug user, where you have here an enhanced spot of CCR5 expressing cells and expression of viral RNA by RNAscope. So now we want to know everything about those brains. And of course, we look a lot at animal models, but what is most important for us is the final subject of interest, that is the human being. And so we want to know the most we can about the environment of the central nervous system in the context of substance use disorders. Of course, our idea is that in the context of substance use, the disease is different. The neurological disorders have a different etiology because of the presence of those neurotransmitters. So how can we access global changes? So the natural answer will be, oh, let's look at the transcriptome, right? However, when it comes to human specimens, we have several challenges to understand things using transcriptome strategies. And this is largely because sometimes, you know, it takes some time between the collection of samples and you know, the post-mortem time period can vary a lot. And to do the transcriptome analysis, we need RNA, and RNA starts degrading almost immediately. So if you take too long to take those specimens, or if the specimens are stored for a very long time, we start losing RNA integrity. And so the value of the specimens for transcriptome analysis starts going exponentially down. So this is a challenge, because then we end up with a very limited number of specimens that can be used for transcriptional studies. So in our lab, we entered in this mission of how can we retrieve information from these brains that are not suited for transcriptome analysis. And this would be important because then it can increase the power of analysis. It can increase the power of association. And we can understand better the diversity of phenotypes that you find in the human condition. So the hypothesis that we developed is that instead of looking at the transcriptome, we could look at epigenomic signatures. And with the idea that these epigenomic signatures, which are, you know, pretty much proteins bound to chromatin on the DNA, right? And so this should be more stable than RNA for us to be able to assess these global changes that are associated with the neurological disorders in the context of substance use. And so we started studying a little bit epigenetic marks that have been in the literature positively associated with gene transcription. And we got into three different possibilities. So as you know, the chromatin is wrapped around the histones, and these modifications on the lysines of the histones can keep the chromatin bound tight together in conditions that preserve the information, and transcription doesn't happen. But sometimes you can have methylation, and then the chromatin or acetylation, so it's a balance of acetylation and methylation, and the chromatin can loosen up in structure. So the architecture modifies slightly in order to open and make available transcription factor binding sites, and then transcription can happen. So we started looking at this tree, that is the histone 3 lysine 27 acetylation, lysine 4 methylation, and lysine 4 3 methylation. These are all positively associated with transcription. And another thing that is can be found in chromatin is the recruitment of RNA polymerase to transcription starting sites. Those also have to correlate with transcription, because those are all events that precede transcription and are more likely more stable than transcription. They are evolutionarily developed to preserve the integrity of the message and the mechanism of writing that message to make proteins. So these are things that we hypothesized would be more stable, and we could look at it. So we started this strategy using chromatin immunoprecipitation followed by sequencing, and we tested all these modifications, histone modifications, and the recruitment of RNA polymerase as well. So the approach then was to test the stability of these epigenetic marks in different post-mortem intervals, and then do this in mouse models that we can control post-mortem intervals, confirm that the whatever epigenetic mark that was more stable in the post-mortem intervals, across post-mortem intervals in mice, would be also present in human post-mortem brain specimens with poor RNA quality, and then test our ability to utilize this strategy to predict the behavior of genes and their functional networks, resulting in useful information to understand what happens to the environment of HIV-infected brain in the context of methamphetamine addiction. So the first part of the question, which is, are these histone modifications and RNA polymerase stable throughout different post-mortem intervals? So what we did in this experiment, which Liana, that works in my lab, says is the most disgusting experiment she ever made. We took mice and we sacrificed them, and we left them at the bench at room temperature for several days. So then we collected the brains at 6 hours, 24, 48, 72, and 96 hours after post-mortem. And we tested the events, the chromatin modifications in 2 housekeeping genes. So these are genes that should be always being transcribed. So positive histone modification should be always there, right? So what we see here, for instance, in actin and GAPDH, is that we have at 6 hours post-mortem, we have a high number of events in both genes and on the promoter of both genes. And over time, this tends to decrease. So 24 hours, you already have a significant decrease in RNA polymerase. Look at this one. In GAPDH, it becomes almost nothing. It's almost like the negative control. So RNA polymerase, based on this, is not a good mark to look in post-mortem tissue. H3K27 acetylation, which is the super enhancer, also we lose it a lot. It's kind of stable, but you can see that the slope is quite drastic, right? And we agree that histone 3 lysine 4-3 methylation, He4-3 methylation, which also we have some loss, but apart from the 96 hours, which might not produce any useful data, we have a more gentle slope. So we chose H3K4-3 methylation to examine in human specimens. So in human specimens here, you can see that H3K4-3 methylation is also the most abundant, oh, I'm so sorry, the most abundant histone modification in the 2 genes of housekeeping genes, actin and GAPDH. And we can detect in both HIV positive individuals that are not MET users and the ones that are MET users as well. So we are good with this. So we can go ahead and test a higher number of specimens. So this is just for you to see how the H3K4-3 methylation looks like when we compare an HIV a no-MET user with a MET user. So they are both HIV positive subjects, but this one doesn't use any drug and this one does. So you can see that on the proximal promoter, you find signal, enhancer signal for this gene, for instance, that is a gene involved in apoptosis, which is present in the MET user, but not in the no-MET user. So I think we can distinguish the differences between individuals. So now we selected several different cases. So we selected 6 cases. So 3 of them no-MET users and 3 that were MET users. They are all treated with antiretrovirals with a variable CD4 nadir, suggesting that they had a slightly different viral load perhaps, but what they all have in common is a poor RNA quality. So this is a measure of RNA integrity, and a value that is below 7.2 is not acceptable for transcriptome analysis, so which means that the RNA is fragmented, and so all of them are poor RNA quality and cannot be used in transcriptomes. So we tested them by ChIP-seq to H3K4, is missing a 4 here, 3 methylation. So here is just for you to see data quality. All of them had a very homogeneous profile, which is good. And we were able to, by analyzing the promoters that have K4 trimethylation, we were able to segregate the 2 groups very nicely. So we found signatures that are exclusive to HIV plus MET negative, and signatures that are exclusive to MET positives. And we found also a large number of genes that are overlap between the 2 groups. And here is just for you to see that the 2 groups have similar activity throughout chromosomes, which is compatible to what is expected. So now, how do we put all this together and transform into useful information? So then we applied our expertise in systems biology methods, which is about using a combination of different strategies to examine pathways, but overall to predict the behavior of networks of genes that are connected in biological processes. So we use Ingenuity pathway. I think many people do that. We also like David Bioinformatics Resources, which is an old resource, but is reliable. We like it. And then we use several visualization tools, including our favorite is Cytoscape, which is a platform for visualization developed in UCSD by Dr. Heidker. And we use as a plugin GeneMania. Our background Reactome and BioGrid networks are also used in this Cytoscape platform. And we use TRANSFAC that is developed by GeneXplain to model transcription factor usage in the context of the differences that we observe. This is interesting and important because several, the epigenetic marks and the transcription factor dynamic, they are interrelated. So many of the epigenetic changes happen because of bridging of available transcription factors and vice versa. Once you have an opening of chromatin, whatever is available in the environment might be likely to produce transcriptional outcomes. And so it's important to learn about this, also to think about ways to interfere with the process. So we have some work that I can come back another day to tell you about, where we took this information and we knocked down the transcription factor to see how important it was, and we were very happy with the results. So anyway, in the human samples, we cannot knock down with CRISPR, but we can predict the behavior of genes of interest. So what do we find now? So we can see that by using epigenetic marks, such as H3K4 trimethylation, we can identify functional annotations and also very specific signatures of interest. For instance, we see that when we compare HIV positive individuals without drug abuse and with methamphetamine abuse, we overlap them, or we compare them. And what we see here in orange color is everything that is increased by the drug, and what is in blue is what is decreased in correlation with the drug use. So we see that methamphetamine brings up several molecular markers that are associated to increased interferon gamma. So what we are looking here is at promoter activity, right? So this is not transcription. This is an increase in promoter activity at the level of the chromatin. And so what we see here is a number of pro-inflammatory functional changes. And in correlation with that, we have a decrease in things that are cognitive. So it's a brain environment, so it makes sense that we may have, in the context of methamphetamine, an increase in inflammation that is bringing down pathways that are associated to cognition. So it might be an effect of neurotoxicity. And we can also identify specific signatures. So we were very happy to see that we can detect changes in promoter activity in several genes associated to the dopaminergic system. So we have enhancement, we have increase in some sets of genes and decrease in other sets of genes. So, of course, because of the RNA quality, we cannot tell if this is really associated to changes in transcription, but very likely because we are looking at proximal promoter activity. But we do have clinical data of all these individuals, so we can attest that there are symptoms that are compatible with dopaminergic disorders. So we are happy with that. We have inflammation and changes in significant changes in promoter activity of genes in the dopaminergic system. So now, how can we now make a prediction of how transcription factors are involved? So now we use the TRANSFAC I use the FMatch algorithm, and when we throw the data in the format of a bed, so you have to throw in raw format. So it's a wonderful thing that it's a not at all biased analysis, because what you have to put in the algorithm is the raw data. So it's totally unbiased. So we can derive a number of potentially important transcription factors. So the way the algorithm works is by aligning or counting the number of transcription factor binding motifs in proximal promoters. I select the proximal promoter part of the story, and then it will count the number of sites in interval sequences. So here we can see that SP1 is a winner with 4 counts per interval sequence. And I focus on everything that is above 2-fold. So it's 2 counts. So we can make a list over here. And when we examine the literature, what's the role of these transcription factors in neurological disorders and in HIV, we find a lot of interesting information. So this can educate us on the next steps. So if we have now models that replicate aspects of the human disease, we can try to knock down these transcription factors to see the relative value or the relative importance of them in producing neurological pathology outcomes. So now we can make a reverse analysis here. So if I get the information that I obtained from TRANSFAC, and I now put it back into the Cytoscape platform, and I use for this another plugin called iRegulon, and I ask the application to identify in my data, in all the genes with transcriptional, with promoter activity, H3K4 trimethylation activity, which give me 20 genes that can be regulated by the transcription factors that TRANSFAC identified. So find me 20 genes in my data that are regulated by these transcription factors. And then I find that there is a lot of overlap, a lot of relationships, related processes, which is very interesting. So it means that there is something really going on here. And now, for instance, genes that are likely to be regulated by ETS2 are mainly with decreased promoter activity. So it can be that methamphetamine is bringing this down. The same here with SP1. So we found changes in genes that are regulated by these transcription factors. So now the question is, can we now fix the problem by using strategies that enhance transcriptional factor activity with a focus on these 2 guys over here? So these are interesting questions to ask. Or the other way around with HOX5 and this other transcription factor over here, where you have enhanced promoter activity. So what can we modify by suppressing this kind of transcription factor activity? So then we have PAX6. These are things with neurological implications. So Now this will inform the next step. So now if I get all this and I put it back into into questioning what are the functional annotations of these gene networks, we get happy because we see that indeed we have regulation of polymerase to recruitment, we have response to drug, which is interesting, we have viral processes, we have even aging. So the hypothesis that methamphetamine may increase the aging process might make sense over here based on this kind of analysis. So it's very central. We can now expand our question and ask what else can we see in this data that would make sense in terms of substance use disorders. And, you know, so we looked at other regions. Now we open not only to proximal promoter, but now looking for H3K4 trimethylation activity in other regions of the genes which might affect genomic architecture. And now we find activity changes in several genes that have consequences to addiction pathways. And even more specific, we can narrow even more in amphetamine pathway. So we also find changes in nicotine pathways, morphine pathways, cocaine pathways. I would be cautious to provide the interpretation of the data because we don't know what else these individuals were up to, but polysubstance use is a problem in human subjects. So this might be something important to look at, and also because of the overlap of addiction pathways that we may encounter. We also identify with a high degree of granularity differences in pathways associated with neurological synapses. And what is most striking to me is that when you compare HIV plus methamphetamine negative with methamphetamine positive, what we see is a drastic downregulation of gene activity in the MET users. So these are all synaptic pathways, right? Long-term potentiation, neuroactive ligand receptor interaction, synaptic, very specific synaptic pathways. So what this suggests to me is that MET use is decreasing synaptic strength. So the pathways that are the gene activity in pathways that enhance or that promote synaptic communication is really going down. And so this might be an underlying cause for cognitive deficits. So just to conclude, I think we are good on time and we may have time for questions. So we can use system strategies to analyze epigenetic data. And this is something that can be very helpful in analyzing post-mortem specimens, in our case, brain specimens. These are strategies that are usually applied to transcriptional data, but we show here that by using these strategies on epigenomic activity data, we can identify with a high degree of granularity changes that distinguish HIV-infected individuals that are MET users or no MET users. So again, demonstrating that we have a different disease. And what else we have here? That these are likely to reflect transcriptional pathways or patterns, but in the human specimens, we cannot confirm that, but in mouse models, we do. And we can predict transcription factor overrepresentation in this kind of data. And this is going to be very important now to estimate biological processes, to estimate potential targets for therapy. And finally, to conclude that we believe that H3K43 methylation can be a very useful alternative to transcriptome strategies to increase the power of association using specimens with long post-mortem intervals and with overall low RNA quality. So just, I would like to thank Liana Bazova, which is the most responsible for this work. We had the help of undergrad students, Anne-Marie from Mesa College, Alex from UCSD, Dr. Ron Ellis, that helped us to select the cases for this study. Mariana Scherner, that is a great science buddy, great discussions. I want to especially thank Dr. John Satterley because he was incredible at providing suggestions and encouraging us to think out of the box and do the things that we have to do to get this data done. Dr. Howard Fox, fantastic discussions, and above all, our funding, NIH, NIDA, thank you so much. And so now I can entertain your questions.
Thank you very much, Cecilia, for your wonderful presentation. Thank you indeed. And as Cecilia mentioned, we are open now for questions. Please feel free to type your questions into Q&A session. So far, we got one question or comment, Cecilia, from someone who is in drug discovery research and asking to mention, maybe to comment how gene expression profile helps in drug discovery. Could you comment something on this?
Yeah, of course. So yeah, this is a wonderful question, and I'm sure that people in drug discovery ask this question all the time. I have a husband in drug discovery, and he certainly looks at me and says, "What?" But it does help because when you introduce strategies, analytical strategies, or when you integrate this data with clinical health determinants, when you use this as a way to predict, let's say, transcription factor usage, you can identify regions, hotspots, right? So, of course, when you talk about a pathology, we tend to treat the symptoms, okay? So when you do drug discovery, you say, oh, I'm gonna treat the symptoms, right? Or you will look for the pockets in the molecule. So you are going to treat at the protein level. But when you are in the step before, it's a discovery step, right? You still don't know which protein you are going to target, right? So you have to figure out what is the functional alteration, what is the biological process that is modified. And then let's say when you have a transcription factor prediction resource, you can perhaps use that as a target, right? So we had a previous work where we were studying effects of transactivation of transcription. And we found several overlapping effects in vitro. So then what we did was to CRISPR the transcription factor and show that this was indeed something important in regulating several of the inflammatory changes. So this is one step. So these are all discovery steps. So when you get to understanding significant biological processes that are modified in the context of pathology, you can predict or estimate what are the important proteins, regulators that in the drug discovery step will serve for you as a target in silico or, you know, in the wet lab. So it's a discovery step. So now we move forward. I don't know if I answered your question, but you can tell me if you want to discuss more. I'm happy to.
Thank you very much, Cecilia. I think it was a very good answer to this question.
I think there is a question, one more question.
Yeah,
here.
right. The question is there any method to study whole genome comparison? If so, will it be sensible? So, because you have done studies of these enhancers, that methylation points mark these enhancers, and question is about whole genome. Maybe
yes, so those are the GWAS, right? I don't know if this is what you are asking, but there are genome-wide association studies, right? Which is something that people use a lot to make comparisons. And then from what I see, what people do is to study genome sequence, right? So then they do the whole genome sequence and they will identify single nucleotide polymorphisms that are more likely to, or that have an enhanced frequency in a given population. So the methamphetamine users or the cannabis users or alcohol dependence. So this exists. From what I see, most of the time you get 1-2 hits. And there is a huge need to integrate this data with a clinical biomarkers to make sense of it. So, of course, these are all different levels of analysis. So you have the genome sequence, which is something you inherit. You have the transcription of the message, right? So the epigenetic regulation is a second level of the problem. So then you have the interference of the regulators and how the chromatin architecture modifies. This is a second level. Then you have the post-transcriptional modification. So it's a layer of different processes that can go wrong or that can be modified by the environment, right? So it's gene plus the environment. I think that's what we learn in school, and it's really not wrong because there is much more that can be modified by your lifestyle or the things you do, the place you live, you know, the kind of life you have, than the genome itself. And so far, I think the genomic studies have given very little information. There is some information. If you get your 23andMe swab test, you can predict a few things, but there is a lot to learn from that. And I think epigenomic transcriptomics and proteomic studies need to be all integrated and also together with clinical data to get to an answer, which might not be very simple, I don't know if I answer, but it's a complicated answer, but it's an answer.
Yeah, thank you very much, Cecilia. Let's go to the next question. Thank you very much for your very interesting talk. What would be your expectation for mass plus only individuals? In other words, what is the impact of the HIV infection of your results?
Yeah, that's a great question. As you may guess, these are very expensive experiments. So at this point, we had to narrow and ask what is the effect of math in the context of HIV. But from mouse models, we know that math has a very strong effect on suppressing gene transcription. Now, transcriptome studies in mouse, we know that math has a massive suppressive effect, not on inflammatory genes though, right? So it's if you look at the overall effect of math is suppressive, but there is this localized effect on pro-inflammatory genes which are enhanced. And in this overall suppression, HIV tends to bring it back. So it's a puzzling effect. So that makes things very hard because for most of the things we look for, we don't see difference from controls. But when we narrow to processes, biological processes, that's where we find the hotspots.
Thank you very much, Cecilia. The next question is more technical, maybe. Someone is asking, where did I get information about software for gene expression profile? Maybe for analysis of gene expression profile, because it is about software for analysis. So where they can get, so where you collected your software? Maybe you just share your experience.
Yeah, my experience. Well, trying first, right? If you are interested in learning this, I would look for a good mentor, and there are several resources. This is, for instance, Cytoscape is a free platform, but it's not really gene expression analysis. It's more like visualization. You kind of know what you're looking for when you get to that point, but also depends on what kind of plugin you are using. If there are several resources. GeneXplain is one of them. They have wonderful resources. I mean, I focus on the transcription factor analysis, but of course they have more stuff. Usually these resources have tutorials so that you can go online and make these short courses and learn a little bit. In general, let's say in UCSD, there is a wonderful bioengineering course. So people usually go for bioengineering to learn these things, but you find a lot online. Also the literature, when you read papers and you see, oh, this guy used this FMatch thing, what is it? So you go look for it. So when you read the papers on systems biology, you find a lot of information, and then you have to just look for it. So how do you find the searching? And,
thank you very much. That's it. Yeah, and for the moment, we do not have more questions, and I think
there is a question here. There is a question. somebody is asking, what is the distance to TSS from ChIP-seq marks in enhanced that you have considered? Okay, so yeah, we did two kinds of analysis. So in the first one, when we showed the functional annotation and the overall dopaminergic thing, we are restricted to 2KB. And but you can open. So when you find a synaptic and addiction pathway changes, it's all over. So it's a proximal promoter, distal promoter, intron, exon, everything. So we can, as long as you know that these modifications in different regions of the gene have different functions. So let's say if you are in the proximal promoter, you are more likely to have a transcriptional result. But on the distal promoter or even somewhere else, you can have regulators and you know, differences in the architecture that can bring things together. So you have to keep that 3D idea in mind. And but you, as long as you know what is the exact function you are interested on, regulation or actual promoter activity, you just narrow.
Yeah, thank you very much, Cecilia. And our time is also almost over. So for all participants, you are very welcome to send us further questions to info@genexplain.com, and we forward all this to Cecilia, and Cecilia would be so kind to comment if you have further questions later on. So for the moment, we are very thankful to our presenter today. And yeah, we just would like to say goodbye to everyone and have a very good day or very good evening.
Thank you so much.
Thank you very much. Bye.
Thank you.
Goodbye.